Yeah, this actually got started using the website LinkedIn. I saw a conversation lamenting that diagnostic companies didn't participate with the International Society for Neglected Tropical Diseases, and I came to the defense of the industry. I'm going to talk about a little bit about our company, EMD Millipore. I'll introduce you to the Muse technology. We're about to release the CD4 system. We're initiating a malaria program. I want to talk about how to work with large companies, how to work with a company like Merck to bring your assays to reality. I'm talking about the Muse and the beginning of a screening technology, but also I'd like to talk to you about confirmation technologies. Real quick, Merck is a huge company made up of a lot of small companies divided into cells. I work in one of them called the Flow Imaging Group, and my job is to look under rocks and find business where it doesn't look like anybody else is there, which fits into tropical diseases. Our sales are split by region, but if you'll notice, 24% of our market is in emerging markets. So we are in Africa, and we've worked there 10 years in the CD4 business. Our products are all benchtop or smaller. I tell everybody we have a desktop flow cytometer. So with that, I'll go on and introduce you to the Muse. This is the Muse. It weighs about 4 kilograms. It's tiny. It can fit in a knapsack, or it could be easily installed into anyone's lab. It runs on electricity. We've spent 3 years developing this product. Recently, we replaced numerous parts in it to make it very, very durable. We worked very closely with the University of Yaoundé in Cameroon, and it's ready for African humidity and African electricity. It comes with an accessory of a UPS, and we're also developing a battery pack for it. It's an economical way to measure the progression of the HIV virus in adults and children. It's a low cost per test, under €3 a test. The big advantage to it is a small sample requirement, and it's extremely easy to use. I tell everybody if you can pipette and operate a laptop, we can train you in under an hour. And it's very environmentally friendly. Essentially, we have a capillary. Behind the capillary is a syringe pump. It pulls 10 microliters of sample up through the capillary where a point of interrogation is. The laser light hits the cells and we count it. So it's a very, very small flow cytometer with 2 colors and forward scatter. Forward scatter gives us the relative size of the cell. We use the 2 colors to do a complete phenotype of the lymphocyte population. So we deliver a total lymph count, a CD4 count, and a CD4 percentage on every sample. Here's where our patents come in. We wanted to improve the ability to count in cytopenic specimens, like you get in tertiary AIDS patients. And with a traditional flow cytometer, and you see a lens on the top, the lens is very distant from where you count the cells. This is to get resolution on the interior of the cells. If you're just looking at fluorescent markers and counting cells at very low levels, you don't need that type of resolution. So we mounted the lens onto the capillary, and we found that we had a huge advantage in counting cells at very, very low levels. And I have examples of this here. So our flow cytometer fits inside a Coke can. It's a very, very small flow system, and we condensed large instruments into a very small, compact instrument. This is an example of a cytopedia, very low count. We found that we can get down to about 10 cells per microliter. Most of the point-of-care CD4 systems are no good below 300 cells per microliter. So we feel that there's hope in seeing the T-cell populations recover with your medical protocol. So the Muse Auto CD4 system will be released. It just finished clinical trials. We're putting our clinical file together, and we should have a CE-IVD in the July timeframe. I ask you to bring it into your laboratories and test it yourself. We sell through evaluations. Our whole motto is the empirical test is best, and we'll go up against any other technology. Our team is just initiating a malaria assay. We've been co-funded by our good friends at Merck Serono. You might want to give them a hand. The goal is to set up a screening and a phenotyping assay for malaria and also now for dengue fever. The ideal outcome would allow 4 to 5 parasites, also the viruses. We have a big concern for pediatric patients. One of our corporate pillars of responsibility is a concern for children. The project is in the early stages. We're just assembling our team, but we've already had some initial thoughts on it. One of the things that we're going to do is have a new indice in hematology. It'll be the percentage of red cells taken up by parasites. I'm not going to go into all the different methods that are out there right now. I'll just say that anything that could be set up on ELISA could easily be set up on the Muse. It'll be faster, it'll be more sensitive, and it'll be more specific than the other methods. It'll also be much more affordable and less time consuming. An average test on the Muse takes 3 minutes. The Muse is designed for ambulatory care centers, laboratories, and clinics. The big advantage to flow cytometry is we have some expertise. We feel like we can deliver it at a low-cost assay, and we think we can do multiple neglected tropical diseases in flow cytometry. So again, any test that's currently being done on ELISA could be easily developed for the Muse. And when we look at a project, we do a technical and we do a financial feasibility. We look at the current methods. We look at the availability of the protein, DNA, RNA antibodies, and reagents. And we estimate how many instruments and how many kits would be needed for each disease. And these are things that if you bring that data to us, it helps us in rendering a decision real fast. I want to say that getting the malaria project started took me 3 years to convince the company. And I'll say that I'm very tenacious, and I hang on there until they say yes. So it's worked out. If we need alternate funding, Cameron here has volunteered to work with me to secure other funds. Merck Millipore is not necessarily going to say no because all the money has to come from Merck. We do look at other partners. We partner with a lot of diagnostic companies. We also partner with, we're willing to partner with charities and other groups. You know, each assay is unique, but a typical assay will take maybe 2 years of product development. Normally what I do is we look, like with malaria, for a partner in Africa that we can work with and do trials with. On the CD4 system, we did close to 5 R&D trials before we finally released it for clinical trials. So we're very, very detailed, and we take our time. We don't want to feel rushed to get something to market. We want to do it right the first time. The other piece of it is the regulatory process. That's something that I can't control, but it usually takes numerous clinical trials before we can release a product and have either FDA 510K or CE-IVD. But we enjoy collaborating efforts, and I want to encourage you to approach us. We are willing to work with you. It's, you know, I'm surprised my competitors aren't all here, but we welcome the opportunity to work with you. We love working with academians, and we love working with other companies. So we're looking at long-term for the Muse, or the Muse series of instruments, as a screening device to monitor recovery, to test cell viability. We're working with malaria, but we're willing to do other tropical diseases and immunology. And we think that an inexpensive screening device, keeping the cost per test at €3 or test or below, will increase your exposure or enhance medicine to all sorts of people. There's two ways to do this. One is portability. Joseph addressed that. And the other piece is the price. We also wanted to talk about the confirmatory device because very few companies address this. We have a new technology called flow imaging. It's capable, it's intuitive, and it's very, very affordable. So imagine that you have screening devices all over the countryside, but you have the big central hospital. You can use something like the Muse to determine which patients would go to the hospital, send the sample to the hospital, and the pathologist can look at it and actually see pictures of up to 10,000 cells in under 3 minutes. And we work very closely with blood cells. We can pull up the different types of, these are white blood cells. It'd be easy to do. We can do complete phenotyping. It reveals very, very much, and this is no more expensive than a standard hospital PCA or a standard hospital flow cytometer. So it's a very, very affordable technology, but it's related closely to microscopy. Somebody said in an earlier presentation, the beauty of microscopy is the ability to do morphology. Well, I tell you, why look at the microscope when the macroscope can show you so much more? And I wanted to, since we were talking about parasites so much, I thought I'd show you some parasites in cells so that you can get an idea of how good this would be as a diagnostic tool. This instrument is a research instrument at this point, but if I can build up enough and generate enough interest, we could develop it into a diagnostic tool. Here's plasmodium in cells. Like I said, it takes up to 10,000 pictures. And so you can imagine a good differential manually is 100 cells. 10,000 cells is going to increase your sensitivity and your specificity. You know, here's Brucei. It's all here. Gandhi-infected monocytes. We've done a classification of malaria. So I think this is the future. Have this in the big account where the pathologist is, and he can make the final decision on the patient. So all I can say is contact me if you'd like to work with us, and we welcome your participation.